Welcome to your first online lecture. This is Environmental Health, and I'm Ben Greenfield. Before we launch into our discussion of today's main topic, which is environmental epidemiology, I wanted to remind you of the key aspect that we discussed in terms of the definition of environmental health last week. For this class, it's important that you understand that environmental health science as a scientific discipline is not just focused on how we affect the environment. Environmental health science as a scientific field is actually considering how our environment affects human health. So, for example, if you're just focused on pollution, things like concentrations of pollution in the soil, if you're focused on things like the health of the ecosystem, so ecological impacts that we've had, this is not by itself within the field of environmental health science. For it to be an environmental health science study, the effects on humans also need to be considered. And so in this class, when I talk about environmental health or environmental health science, you will see that I'm almost always referring back to patterns in terms of our impacts to the environment and how these can in turn affect our health and safety as people. And I also want to draw your attention to the fact that in exams, you'll be expected to understand this definition as it occurs in this class. You'll be expected to be able to use this definition in order to identify whether or not something is an environmental health science problem. So now let's talk about today's topic, environmental epidemiology, in the broader context of the field of environmental health sciences. Environmental health sciences typically includes 4 different topics. By some definitions, there are others, but for the purposes of this class, you can think about environmental health sciences as incorporating environmental epidemiology, toxicology, the study of poisons or things that humans are exposed to that could potentially cause toxic health effects and what those health effects are, exposure assessment, which is the measurement of our rates of exposure to different things in our environment, and integration of these 3 things, environmental epidemiology, toxicology, and exposure assessment, into risk assessment, which is essentially the evaluation of the extent to which some aspect of our environment is causing health effects or the risks, the amount of health effects or risks to human populations. So over the next series of lectures, we're going to be evaluating these different topics. And for this one, we're going to be talking about environmental epidemiology as a field. For this first online lecture, I have an assignment that I want you to do. Basically, throughout the lecture, I'm going to be posting questions in blue. The questions will pertain to the information in the next few slides. What you should do is take notes on that information in order to be able to answer these questions. In class, on the assigned day, I expect you to turn in your notes. I will look at them and return them to you. The goal here is to make sure that you're not only watching the lecture, but getting the main points of information that I want you to learn. So epidemiology is a broad field, and of which environmental epidemiology is a subspecialty. And so before we get into environmental epidemiology, I wanted to provide some insight into some of the main aspects of epidemiology itself. First of all, we're going to cover the definition of epidemiology. We're going to discuss a couple of main concepts about when epidemiology is used and how it's used. I'm going to talk about the Bradford Hill criteria, which is a set of historic guidelines for what constitutes strong evidence in the epidemiology framework. And finally, we're going to discuss the issue of ethical concerns and touch on that briefly. So here's the first of these questions. So you can go ahead and pause it and write down the questions. And like I said, then you should take notes on the answers to these questions from the next couple of slides. The questions for the next few slides are, What is epidemiology? Actually, this is just for the next slide. What is epidemiology? Why should we study epidemiology? And who uses epidemiology data? And these are some of the main concepts in epidemiology. Epidemiology is the study of the distribution and determinants of health and disease in human populations. Let's go through that for a minute so that we can make sure we understand this definition. When we say it's the study of the distribution of health and disease in human populations, we mean that it studies things like spatial patterns in where diseases are happening, say in which states, which countries, or which locations, and also patterns in time. Are diseases increasing or decreasing over time? Are they showing seasonal patterns? Things like that. Determinants is just a fancy way to say causes. And so part of what epidemiology is doing is trying to just study the things that cause health or disease in human populations. So for example, what causes lung cancer in human populations? Or what causes people to have a general good cardiac health, for example, low blood pressure, things like that. Key to all of this is the part about human populations. Epidemiology is the study of people. An epidemiology study, by definition, has people as its study subjects. So if you do a test on laboratory rats and look at, for example, whether or not exposing them to cigarette smoke leads to lung cancer in those rats, that is not epidemiology. However, if you looked, for example, at the rate of lung cancer among smokers and compare that to the rate of lung cancer among non-smokers, that would be on better people. That would be an epidemiology study. The reason we want to study epidemiology is ultimately because we want to figure out ways to prevent disease. Disease is not something that we want, right? We don't want people getting things like lung cancer. And so in order to prevent lung cancer, we want to understand what exposures could cause that disease. So that's the idea. What are the exposures or determinants that cause a disease? And with that information, we can potentially provide prevention. Epidemiology stems fundamentally from the work of an old scientist, historical scientist named Popper. And the idea is basically that when you look at whether something A is followed by something B, this helps you to form a hypothesis that A causes B. So in our example about lung cancer, right, if we observe that people who smoke later in life tend to get lung cancer, then we could develop the causal hypothesis that smoking causes lung cancer, which we could later prove or disprove. We could later disprove it by further testing. And this stems to the Hill criteria. And these criteria will be talked about in the next slide. But these are a set of criteria determining how strong the evidence is in epidemiology studies. The other important use of epidemiology and why it is important is that the criteria evaluating different epidemiology studies. And when epidemiology studies achieve these different criteria, regulators and risk assessors, so various managers, can use them to evaluate causality. So back to our smoking example, by critically evaluating epidemiology studies, regulators and risk assessors may determine, regulators in particular, may determine that there should be some anti-smoking ordinances, such as, for example, the ban on smoking at the SIUE campus. Our next question is, what are examples of the Hill criteria? So the Hill criteria were developed by a researcher named Bradford Hill in 1965. And the idea behind these criteria is that when you're studying any type of biological health outcome, especially among humans, you would want to have an amount of strong supporting evidence in order to develop confidence in your conclusion. And so he developed a set of different criteria, some of which I'm going to describe here. The first one is that the exposure must precede the disease. Sounds pretty basic, but the idea being that if you can establish what's called temporality, or the exposure being before the disease, you have a higher amount of confidence in your conclusion that the exposure likely caused the disease. This ends up being very important in epidemiology study designs, as you'll see later. Those studies that enable you to look at exposures before the disease allow a much greater degree of confidence that the exposure was responsible for the disease. Another one is consistency. That's the next criteria. Consistency is the idea that when you look among different studies, different populations of people, different areas or studies done at different times, you will consistently see the same health outcome in response to a particular exposure. So, for example, cigarette smoking causing lung cancer. There's a huge amount of consistent evidence across many different populations that cigarette smoking oftentimes leads to lung cancer. The next criteria was large effect size. So all else being equal, if you do a study of a particular exposure and then the health outcome or disease, did that exposure cause a very strong disease effect, or was it a very small disease effect? The stronger the effect size, the stronger the relationship between the exposure and the disease, the more confident we are in the fact that it was a cause. Another one is a positive dose-response relationship. So here the idea, right, is the dose response. So as you increase your exposure, do you see an increase in the rate of disease, right? Do you see, like, as exposure goes up, does disease go up? So here would be your exposure, and here's your disease. Do you see a strong increasing relationship here? If you do, that increases your confidence that the exposure was responsible for the disease. If the relationship is more like this, and as the exposure is going up, the disease is not going up, then you don't really have confidence that the exposure is leading to the disease. So if you have evidence of the positive dose-response relationship, you have more strength of evidence of the exposure causing the disease. The next one is biological plausibility. Is there some kind of underlying biological mechanism, say something like DNA damage, or some evidence of weakening of immune system function that you can show, demonstrate evidence of for that exposure causing that disease? So those are some of the Bradford-Hill criteria. And now we are going to move on to the issue of ethics and the importance of ethics in epidemiology. And so your next question that I want you to answer, it's going to relate to the next slide. And the question is this: what ethical principles of epidemiology did the Tuskegee syphilis study violate? How was the Tuskegee syphilis study unethical, specifically in terms of principles of epidemiology? And then the other part is, what other ethical problems were there with the Tuskegee syphilis study? There are actually a number of ethical concerns in epidemiology, and this is a topic that we're touching on in this class, but that I want you to be aware of because it's really important. In epidemiology, we have to consider the concept of informed consent. Informed consent is the idea that people are both informed that they're participating in a study, so we tell them about it, and that they also consent or agree to participating in that study. Now, the consent is really important in terms of making sure that they are up for being part of the study. You've got to make sure that they approve of it. But informed is equally important. They have to know what they're getting into. And that if you don't achieve informed consent, you don't do this prior to doing the study, you run the risk of exploiting your study population, which is certainly something that we do not want to do. Let's talk about an example. The example that I think is one of the most well-established and disturbing examples of a study that was highly exploitive and did not achieve informed consent was the Tuskegee syphilis study. Now, interestingly, the Tuskegee syphilis study initially began in a way that was not really exploitive. Basically, what they did was they enrolled the researchers enrolled a number of African American men. This was in the South. They were enrolled in a study to examine the potential health effects of syphilis over the long term of their lives, over a period of time. And I believe that they included men who had syphilis and men who didn't have syphilis. And you might say, well, why was it not initially an exploitive study? And the reason I would say that one could argue that initially it wasn't exploitive was that these men got syphilis at a time when there was no known treatment for syphilis. So just the act of studying them, and they were provided with health care, and as participants in the study, they were given access to health care and some, medical treatments and given information. So they benefited for their participation, and there was no treatment to syphilis at that time. So at that time, it wasn't really a major ethical concern. But the study became highly unethical when, after a period of time, penicillin was discovered as an effective treatment for syphilis. And so, given a dose of penicillin antibiotics or related antibiotics, syphilis could be treated and effectively cured. The researchers decided not to give penicillin to these subjects. They also did not inform them about penicillin as a possible treatment. And this actually, it wasn't just the researchers, it was also medical doctors in this area. They decided not to give medicine to help treat these subjects. Now, the arguable reason why they said that they did that is because they wanted to look at the long-term chronic health effects of syphilis on a population. One might consider that that could have some beneficial information in terms of understanding the disease progression of syphilis in a population. However, this was certainly not worth the health effects on these poor men that essentially were not given treatment. They were withheld treatment, and they also were not informed about this. So this is an example of why informed consent is so necessary. Could you imagine anyone who was told, we are going to withhold treatment from you in order to be able to study the effect of a disease on you? Do you consent to that? Do you agree to that? No one would say yes, right? So it's really a horrible thing. And so it's actually become a big part of any type of human research, including epidemiology, that informed consent is required. People need to be told about the study and need to agree to participate in the study, having been given sufficient information so that they can understand the study on their own terms and ask any questions and then agree to the study. The other ethical concern that was really violated with the Tuskegee study is the fact that generally, when doing any type of research on humans, it's a given that the benefits of the study should outweigh any potential harms. And further, that there should really not be harm to the study subjects at all of participating in the study. The study subjects should have a benefit, or at least not be harmed by participating in the study. And allowing that to happen in the Tuskegee study was really horrible. So just a kind of an important example of ethical concerns in epidemiology. So now we move on to the next question. This is your next question that you should provide a written answer to in your notes that you're going to turn in. What is environmental epidemiology? Environmental epidemiology is epidemiology, but studying environmental factors. So basically, in environmental epidemiology, we are interested in evaluating environmental exposures and whether or not they cause diseases or health conditions. So these could be health benefits, such as low blood pressure, or diseases, such as our example of lung cancer. So basically, environmental epidemiology is epidemiology, the study of humans and the causes of their diseases. But it uses the methods of epidemiology to study environmental exposures. So here's the next question, and you don't have to write this down, but you should try to answer this question on your own. You might want to pause and look at the definition of environmental epidemiology and try to understand it that you just wrote. And now try to answer this question. Which of these are within the purview of environmental epidemiology? Or which of these studies fit within environmental epidemiology? If we were studying asthma in long-haul truckers, that's the first one. That one is an environmental epidemiology study. Asthma is a health outcome, and the occupational environment of long-haul truckers is going to include air pollution. So we have air pollution as a potential cause of asthma. And because it's an environmental thing, that is the occupational environment, and a health outcome, and we're studying humans, that's environmental epidemiology. Here's the next one. Birth defects for mothers exposed to secondhand smoke. So pause the slide for a minute and ask yourself, is this environmental epidemiology? Have you decided? Yes, this is environmental epidemiology. It's environmental epidemiology because the secondhand smoke is in the mother's environment, and in the child's environment, the sort of the prenatal environment of the child. It is an environmental factor because it's not within the control of the mother or the child whether they're exposed to secondhand smoke. It's part of their environment that they cannot control. Here's the third one. Liver disease versus alcohol consumption. Is this an environmental epidemiology study? Pause the slide for a minute and try to answer. See if you can figure out whether it is or not, and why or why not. Did you get it right? No. Was your answer no? If it was, then you got it right. Alcohol is not an environmental exposure. It's within an individual's control. It's not in their surrounding environment. People would choose whether or not to drink alcohol. So alcohol related to liver disease and the consumption of alcohol versus liver disease is an epidemiology subject, but not environmental epidemiology, because this is within individual control. And here's our final example. Cancer in towns near Chernobyl. If you don't know what Chernobyl is, it happened before you were born, most likely. This was a major meltdown of a nuclear power plant in Russia. And when this meltdown happened, large amounts of radiation were released. And many of the towns near there was concern about people getting increased levels of cancer. So the study of this question of whether cancer is increased in towns near this radiation meltdown, is that environmental epidemiology? Pause and consider your answer. The answer is yes, because here we're talking about the surrounding environment that people live in and the radiation levels in that environment. It's the outside environment that they're exposed to, but it's not within their control, and therefore it's environmental epidemiology. So now let's talk a little bit about epidemiology methods. Epidemiology uses observational data, and this is especially true for environmental epidemiology, because we can't, say, intentionally expose people to pollution, to, say, smoke or air pollution or anything like that, and see the impact on diseases. We couldn't expose them to elevated levels of radiation intentionally. So instead, we have to observe the variation that's occurring in the surrounding environment and relate that to the health outcomes. There are three general kinds of epidemiology methods. These are descriptive studies, And so an example of a descriptive study is simply evaluating the rates of injury or illness for different human populations and seeing how it varies across different professions. So, what's the rate of injury or illness for construction workers versus, say, the rate for nurses? Analytic studies. And so, an analytic study is when you're actually examining relationships that are occurring naturally and evaluating whether or not there seem to be relationships. So, an example here, which is described in your book, is the cholera outbreak that was occurring in London near a particular area where John Snow evaluated and analyzed the spatial patterns and determined the 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 cholera was elevated right near a particular water sources. So the Chimako study, which is a study that you're going to be reading about, this is a great example of an analytic study. This study examines exposure and effects in a particular population over a long period of time and analyzes those relationships. And so we are going to think about, and you're going to be reading about and discussing the methods of the Chimako study and using that as a great case study example of an analytic study. Here's the next question. Define the three general kinds of epidemiology studies and explain the difference between them. The kinds of epidemiology studies include the following. The first type is called a descriptive study. And in a descriptive study, it's basically describing a disease, things like the patterns in space or time of the disease, but it doesn't do any formal hypothesis testing. It doesn't evaluate formally whether there's a particular factor that might be causing that disease. The next type of epidemiology study is called a correlational or ecological study. So the ecological study is defined in epidemiology in a different way from in, say, ecology. The definition of an ecological study is that it's conducting its analysis at the group level. So what this means is you're looking at groups of people and say the disease rate among those groups and the exposure among those groups and comparing one group to another group. So an example might be comparing different countries or comparing different cities. That is a very much of a weaker kind of study design than the next type of designs we're going to talk about, which study individuals. So studies that study individuals, in general, can tell you whether or not individuals with these risk factors might get these diseases. So basically, the point being that the correlational or ecological study is examining the relationships between exposure and disease at a group level. And the third type of study design, and the one that we're going to focus more of our attention on, is what's called the analytical study. There's another term for this, the etiologic study. You don't need to worry about that very much. In the analytical study, it's testing specific exposure disease hypotheses on individuals. So what does that mean? It means that basically the researchers are looking at some kind of exposure and some kind of disease outcome and looking across a whole bunch of people. And in each person looking at, did they have the exposure and did they get that particular health outcome? This being in contrast to the descriptive study, which doesn't characterize the relationship between exposures and disease at all, or the ecological study, which is only looking at exposures and diseases between groups. So an analytical study includes two types of studies. One is the one that we're going to focus most of our attention on is referred to as the observational study. So these are basically, we could think of them as natural experiments, types of studies where we're kind of looking at patterns over time or patterns among different individuals and their exposure and their disease outcome. The other type is called randomized clinical trials. And so in this type of analytical study, basically, um, we randomize who's getting the exposures. We decide beforehand to give the exposure to certain people, and those people are selected randomly. This is oftentimes very useful in things like testing drugs or testing, different types of treatments for disease. Randomized clinical trials are typically not used at all in environmental epidemiology because we can't randomly say we're going to give certain people something like exposure to some pollutant or something like that. We wouldn't intentionally do that. That would be unethical. However, there is one way that randomized clinical trials can be used when uh, looking at environmental epidemiology, and that's the idea of a randomized intervention. So this is to selectively choose some people in the population that you're going to remove an environmental exposure from, i.e. try to make their lives better, and see how that positively impacts their health outcome. One of the things about the randomized clinical trial is that it eliminates what's referred to as confounding. Confounding is an issue that we may talk about later, but basically, randomized clinical trials, when they're done, they have greater confidence in the interpretation than these observational studies, which are more common in environmental epidemiology. So now your next question is going to be to identify the four main kinds of analytical study designs in epidemiology. The first kind is referred to as the cross-sectional study. And we're going to describe this further, but the basic idea in the cross-sectional study is that we're taking a snapshot in time and looking at the exposure, and we're looking at the disease outcome at the same time. So that's the first design, the cross-sectional study. The second one is referred to as the cohort study. And in the cohort study, the main thing about the cohort study is that we enroll a bunch of people in the study. Here's the individuals. And then we follow them over time. And we see, based on whether they had an exposure, how likely they were to get a particular health outcome. The third type of analytical study, observational study design that is used in epidemiology is the case control study. And in the case control study, in that study, the way it is we identify a bunch of people who have a certain health outcome. Those are the cases. And then once we've done that, we select a bunch of people that are similar to those people in many respects, but they don't have the health outcome. So these would be people who don't have cancer. And then we look at their exposures after that and see whether there's differences in their exposures. So that's the case control study. So let's look at these each in a little more depth. So first we begin with the cross-sectional study, which again, we're not looking at changes over time or before or after scenarios. We're looking at everything at once in terms of the exposure and the health outcome. So here's the next question. I want you to explain what is unique about the cross-sectional study. So that's the first part. Then I want you to say what are the main weaknesses in the cross-sectional study, and when is the cross-sectional study used most often. So the key trait of the cross-sectional study is that it measures exposure and disease at the same time. We're not basically looking at the change over time in a disease, or we're not looking at a disease and then going and trying to estimate the exposure previous in time. We're measuring everything at once. So here's an example of a cross-sectional study design. If you were to look at the metal concentrations in the placenta at the same time as childbirth, right? And so when a baby was born and you looked at the birth outcome of that baby, say for example, how large they were, how much they weighed, and compare that to the concentration of metals in the placenta, and you collected all those data at the same time, that would be a cross-sectional study design. The big difficulty or problem with the cross-sectional study is you can't determine whether the exposure preceded the disease or whether it was before the disease. So if we remember back to the Hill criteria, we had this idea of basically a stronger evidence is going to be if you see that the exposure preceded disease, then you can say that the exposure was more likely to cause the disease. But in cross-sectional studies, you're measuring everything at the same time, so there's no way to do that. As a result, the cross-sectional study is a much weaker design than both the cohort and case control studies, which we'll talk about next. Because you can't separate out which caused which. It's possible if you measure at the same time that the health outcome might have caused the exposure, or that they both might have been caused by something else. The point being that you don't have that temporality to draw back on. So the thing about it is that the cross-sectional study is often the only study design that's possible. So we may not have the resources, we may not have the type of data where we could do the cohort or case control study. That said, the cross-sectional study still could provide valid results, especially in terms of developing hypotheses about causes, which would then be confirmed later in cohort or case control studies. So finally, the main use of a cross-sectional study, or when they're often very useful, is when the outcome is a subclinical outcome or an asymptomatic disease. And the reason that that's the case is because the cross-sectional study isn't that costly, you can basically, when people are going in to see their doctor, you could ask if they would be willing to participate in a study based on some data about a subclinical outcome, say something like a certain kind of bacteria in their nostrils. And that bacteria could potentially cause infection, but it hasn't caused infection. You could take a nostril swab and see whether or not they have that bacteria. And then you could do a survey with them and ask them things like whether they have any risk factors from that bacteria. How often do they shower? Do they eat sushi? Do they, if it's in kind of undeveloped areas, do they live in an area with dirt floors? Things like that. So that is an effective time to do the cross-sectional study because you can kind of quickly gather these survey data from a bunch of people. The next study design that I want you to describe is the cohort study. So now I'd like you to explain what a cohort study is. And I also want you to answer these questions. When is the cohort study most useful? What are the strengths of a cohort study? And it has many strengths. And also, what are the main challenges of the cohort study, which I'm going to talk about now. So here's a graphical image of a cohort study. And the main thing about the cohort study, the main characteristic of it, is that in the cohort study, you're going to have a group of interest and a comparison group. Or basically, you're going to have a bunch of people that you're going to enroll in the study. You're going to put them in the study before you know anything about their health. And they may not even have, they shouldn't really have the health outcome that you're interested in. Typically, you're going to enroll them based on exposure. So for example, you might enroll some people who smoke, and you might enroll some other people that don't smoke. So the idea is that you're enrolling based on exposure. Then you follow everyone in the study over time, track them over time, and see which subjects develop the health outcome, and see whether the smokers, for example, were more likely to develop this health outcome. So maybe we could say it's lung cancer than the non-smokers. So to summarize, the definition of the cohort study is that you have some kind of focus and you're going to follow people that are exposed and non-exposed over time, and then you're going to compare them over that time period in terms of their health outcomes. So this is the main thing, is you're going to enroll people based on their exposure, and then you're going to track them over time and compare their health outcome later. So this type of study design is most useful when you have rare exposures and common diseases. And the idea here being, right, if you have a rare exposure, let's say it's something like, well, the great example of the cohort study that we're going to be looking at much further is the Chamaco study. And the exposure here was in utero exposure to agricultural pesticides. So these are unborn fetuses whose mothers are working in the fields and are therefore exposed to elevated pesticide concentrations from agricultural activity. The vast majority of the population does not have that exposure. So it's very useful to have a cohort study in this case because you can specifically enroll these people in the study and then track them where otherwise you wouldn't be able to find these people. And then it's good for common diseases because as you're tracking those people, you may only have 200 people in the cohort. So if it's a very rare disease, say something like mesothelioma, which is extremely uncommon in the general population, those people would not have very many of them in the cohort, would not enough of them in the cohort would happen to be able to be confident about any patterns you see. So there are two kinds of cohort studies. One of them is the prospective study, and this is kind of the traditional kind of cohort study that I've been describing. And the idea here is that you're enrolling people in the study at the very beginning, and then you're tracking them over time moving forward and looking at the health outcome. So the prospective cohort study is really kind of the gold standard in terms of observational epidemiology study designs, because basically you have no reason to think that these people are going to, you have no way to estimate the health outcome when you first enroll them in the study. So there's very little potential for bias. You're going to be tracking the health outcome after you've looked at the exposure. Then you also have something called the retrospective cohort study. And basically, the idea here is that you're going to select a bunch of people based on their exposures in the past. You're going to estimate those exposures, and then you're going to start tracking their health outcome in the present. And but the basic idea being, again, right, you're going to follow exposed and non-exposed people over time and then compare them. So the main strengths of the cohort study are that there's very low evidence of bias, low bias. And there are a number of reasons for this, and you can see them in the book. But one of the biggest thing, like for example, is like let's say you have a disease that might be difficult to recall. Well, you could have all kinds of biases that people are more likely to remember those diseases if they had a certain exposure than they didn't. But because you're starting with the exposure before you start looking at the disease, you have no reason to expect a bias in how they're remembering it. The biggest challenge in the cohort study, especially for the prospective cohort study, is that they're very costly and logistically challenging. You have to enroll all these people in the study, and then you have to track them over many, many years. Each year you do the study, you have to keep track of all the data. There's a lot of coordination. You have to keep track of the people and make sure you don't lose them. So this adds up to a lot of money and logistical challenges. So now we move on to the next study design. And remember, by the way, just to remember, right, you can pause and replay these things in order to be able to answer these questions. So your next questions are to explain, first of all, what is the case control study? When is this study design most useful? And what are the main drawbacks of the case control study? So here's a graphical depiction of the case control study. And the main difference between the case control study and the cohort study is that in the case control study, you are going to be selecting participants based on their health outcome, or their disease. You're not going to be selecting them based on their exposure. That's the cohort study. So you're going to be looking at cancer patients and you're going to be comparing them with non-patients, for example. This is an example of a health outcome, right? I better not try to draw this. So I'm talking, the key word here is health. You're going to be enrolling people based on their health outcome. So that says outcome there. Okay, so it's not working out so well. That's what makes it a case control study. You're going to enroll them, and then you're going to look back at their previous exposure history, try to estimate what their exposure was prior to that. So, for example, were the cancer patients, were they more likely to work in certain types of jobs that had a lot of exposure to metals, for example? Were they more likely to be in occupations like metalsmithing, welding, auto mechanics, working in foundries, things like that, as compared to the control or comparison group. So that's kind of what you do, right? You compare the people who have a health outcome to the people who don't have that health outcome, and then look at their past history and compare it. So to summarize again, the case control study is the opposite of the cohort study, right? We are looking at the health outcome and separating out your study population based on their health outcome. First, you're going to select people that have this health outcome, say cancer, and then you're going to select a comparison group or the controls that don't have the health outcome. In contrast, the cohort study, you're going to be selecting people based on whether they have a certain exposure or whether they don't have that exposure, say, like I said, pesticide exposure in agricultural settings. The case control study is really good for rare diseases and common exposures. And so we think about this with both the cohort and case control study. The idea in these study designs is that you are identifying the study population that you're interested in. And so let's say that you have some really rare disease, right? Again, like say mesothelioma, that's a disease that's turned out to be from asbestos exposure, or certain types of childhood leukemia, which are very, very rare. It's not efficient to just kind of look at the general population, or it's not efficient to enroll people based on exposures and see whether or not they have that disease. But it's very good if you can go select a bunch of those people as your cases that have the disease, and then you can compare those cases to your control population that don't have the disease. It's very efficient for doing that. It's also good for common exposures. It's not good for rare exposures because, again, your study is going to have a bunch of these people with these diseases and without them, but it may not have enough to find rare exposures. Here's the main definition of the case control study. Again, you start with the diseased and the non-diseased groups. Then you look back in time and compare. And what you're comparing is the odds of exposure in each group. And the statistical measure of this is referred to as the odds ratio. So how likely was each group to have this exposure? So case control studies have a number of drawbacks in comparison to cohort studies. Again, like I said, the cohort study is really the gold standard. Case control studies are vulnerable to various types of bias. People may misremember what their exposures were if they suffered from a disease. You know, a really good example of recall bias is when parents' children were diagnosed with autism. The parents would be more likely to remember that those children were vaccinated in the recent past than parents who weren't diagnosed with autism. And so it's been actually very well established in the medical literature that vaccination does not cause autism. There's no evidence that it causes autism. Yet it would be a big risk in a case control study design that if you talk to a bunch of parents whose children recently had autism, recently were diagnosed with autism, they say, oh, well, you know, we did have this vaccine fairly recently, whereas the other ones might not remember that. So that's recall bias. Disease subjects recall things differently. It's also very difficult to choose representative controls, right? So let's say, for example, you have these children with childhood leukemia, right, as a potential exposure. How do you choose the comparison children? Do you choose children that, you got to find children that might be from the same general population? Are they the same age, same race? And this type of thing of how to choose those representative controls is a great challenge. The other thing is that it's very hard to measure past exposure, right? This is a real challenge with the case control study, that if you're trying to go back and estimate things like how much pesticide exposure, how much exposure to metals or something like that, that's hard to do because you don't have that past data. But if these studies are well designed, they should give the same answer as other types of studies, such as cohort studies, if you're able to remove all of those biases. So now we're going to switch gears and talk about the different types of bias in epidemiology. So I mentioned one of these already. I talked about recall bias. But now I'm going to talk about three major kinds of bias. And I want you to say what they are, and I want you to describe each one. So first, I want to talk about bias in general. Bias is one of the biggest challenges in any type of studies, but it's an especially large challenge in environmental epidemiology. The reason that it's a challenge is because these are observational studies. And so there's always the risk that you could be observing patterns that have relationships that are incorrectly estimated. And that if you don't correctly correct for this, then the relationships that you look at the relationships that you're looking at in your study may not actually reflect the underlying relationships in the population. So what are the three main causes of bias? Or the ones I'm going to talk about. One of them is called confounding. And in confounding, this bias is what's the distortion of the exposure-disease relationship by some third variable associated with both the exposure and the disease. So here's an example, right? If welders smoked more than non-welders, then welders would be more likely to have things such as lung cancer. And we might, if we didn't measure the rate of smoking, we might incorrectly conclude that welding is a big risk factor for lung cancer. So in this case, the smoking is a confounder of the relationship between welding and lung cancer. The other way to think about it, and this is typically how you think about confounding, is graphically. Here we have welding, right? And here we have lung cancer, right? And we might be interested in, is there a relationship between welding and lung cancer? But if in fact smokers are more likely to be welders and they're causing lung cancer, then this would cause a bias in this estimated relationship between welding and cancer. So here the smoking is a confounder. This can be adjusted for using various types of statistical methods, such as stratification or multivariate analysis. Another kind of bias is information bias. And in information bias, this is where you are basically misclassifying or incorrectly measuring some exposure. And this can bias results, either make the result bigger or make it smaller, which is biasing it towards the null. So the example I was talking about before was recall bias. Recall bias, right? So that says bias. And so the idea here, right, this example where if people's children were diagnosed with autism, they may be more likely to remember having had a vaccine in the past year than people whose children were not diagnosed with autism. This would overestimate the actual relationship between autism and vaccines when there might not be there's not a real relationship, but if you have this kind of recall bias and you weren't correcting for it, it would lead to a false conclusion. So this information bias is a real challenge, basically, with recall bias being the best example. Finally, we have selection bias, and this is the idea that a study population was selected in some non-representative way. So the exposure-disease relationship in the study does not reflect that in the general population. The idea being basically that the study population that you chose is not similar in its attributes to the overall population, and so what you see in that population is not actually valid for the overall population. A really good example of this is referred to as the healthy worker effect. And the healthy worker effect is basically it turns out that people who work tend to be healthier than people who don't work. And you might therefore conclude that work is good for health. You might also incorrectly say that certain jobs that are actually harmful to health, things like, for example, working with welding or something like that might actually be helpful to health. People might be healthier, but it's actually just the fact that all those people who couldn't work were less healthy to begin with. They dropped out of the employment population. Some of them may have been former welders who got sick. Some of them may have been people who were too sick to work because of other exposures. And so this means basically that the worker population is not representative of the general population and can lead to a lot of bias. And unfortunately, selection bias cannot be corrected for in general analysis. And the final question for the lecture is related to the evaluation of clusters in epidemiology. So what are clusters in epidemiology, and why would we want to study them? So what a cluster is an elevated number of cases in a limited area over a limited time period, suggesting a common exposure or cause. And so basically the idea of studying clusters, we're talking about disease clusters. And these disease clusters effectively are where you have a bunch of disease cases in a particular area, and you think that there might be some reason for them, such as some environmental cause. And so this kind of introduces another area of environmental epidemiology that I'm just kind of introducing you to, which is the idea of spatial epidemiology, the examination of spatial patterns of diseases or health outcomes in the environment, and using that to make some kind of estimates of what might be the causes of the disease. So evaluating environmental clusters is very challenging. It's difficult to establish the geographic or temporal boundaries of different areas. Sometimes you may be looking at a broad disease, such as cancers or something like that, and the different subtypes might have different causes. And oftentimes there's a low number of cases in a disease cluster, and as a result, it's difficult to know whether or not this is just kind of random variation or whether there's actually some kind of environmental cause. It's generally less difficult to look at occupational clusters. So the challenges of environmental clusters are listed here, but with occupational clusters, the workplace is a defined boundary. And so, for example, you might have a bunch of people that are working in a particular coal mine, and they might exhibit higher rates of, respiratory disease or something like that, or people working in mining in general. It's very clear that either they worked in that place or they didn't, and that you could associate the workplace with the disease more readily than environmental clusters. Here's an example of kind of a classic environmental disease cluster. This is a trichloroethylene, which was a chemical that's found in that was polluted into some ponds in this area of Massachusetts. And some of you may have seen a movie called A Civil Action, where basically John Travolta plays this lawyer who is essentially noticing a cluster of more of a certain type of cancer in the area around where a trichloroethylene exposure was higher, and trying to basically sue the owners of the company that were responsible for the spill of this compound into the water system. And so this kind of disease cluster was used as sort of a basis for evidence of kind of environmental pollution, an environmental health problem of the pollution causing a lot of health impacts to the people. Now, you can even see from looking at this data some of the difficulties of evaluating clusters spatially, right? So you see a higher clump of these disease cases here, but you're really not sure, was that just kind of due to sort of random variation, because you can see little clumps in other places too, or whether it's specifically associated with the source. And so epidemiologists with more advanced training spend a lot of effort developing statistical analyses of these kinds of clusters and their associations with environmental patterns as a kind of a part of kind of the geographic aspect of epidemiology. So that concludes the lecture part, the online lecture, and having taken some notes on all of these different questions, you should now have a really quite solid understanding of some of the main fundamentals of environmental epidemiology. And I just want to remind you that the textbook covers all of these topics in more detail, and so for any part that you found that was unclear in what I was presenting, I encourage you to look again at the reading that you did in the textbook to find out more information about it and kind of develop further clarity.